Dear students, welcome to this session. So in the last class we have discussed about the microbes in household products. What are the products we have obtained? The various dairy products, the duff fermentation, etc. Now today in this class we have to know something about the microbes in industrial products. What is their role? What are the different products obtained through industries? And that is what's called microbes in industrial products. So microbes are normally used to synthesize a number of valuable products for human beings. For example, the beverages, the drinks, the antibiotics. Then we have the enzymes, the organic acids, also amino acids, the vaccines. Then we have pharmaceutical drugs. And these are all the different products what we are getting through industries. That's why it's being included under the microbes in industrial products. So here I wrote also, microbes are used to synthesize a number of products. All these products are valuable to human beings and they are also responsible for human welfare. Now what are the products I mentioned about? The beverages, the antibiotics, the vitamins, organic acids, amino acids, the biofuels, single cell protein, what I discussed already, the enzymes, different enzymes, then the steroids to treat the diseases, vaccines also the pharmaceutical drugs, the medicines, etc. All being produced because of the effect of microorganisms. So if you want to get in small amounts, we can just get it. Suppose you want to produce in a large scale, we need to grow the microorganisms, the biomass, in a large vessel. The name of the vessel is called fermenter or bioreactor. If you want to produce large scale, that is actually, further you have to grow the microbes in a large vessel. The name of the vessel is called what is known as a bioreactor or fermenter vessel. So this fermenter vessel is nothing but actually a closed vessel with some adequate arrangement. What for? Aeration, agitation of what is called actually, there is the products, what we introduce, what is called the reactants, and also pH control, temperature control, and drain or overflow vent to just actually remove the waste biomass or what is called the cultured microorganisms along with their products. So it's a simple vessel, just like you see, imagine that one is like what we call a washing machine. There is a rotator, there is what's called the agitator, it is rotating constantly, agitating the materials. Oxygen is being provided through aeration. We need a constant pH, normal alkaline pH, the temperature for the formation of the various bioproducts, otherwise there will be what we have denatured. So all these things are there in the fermenter vessel. I will give that a picture in the video, you see that one. I will also show just a simple one in the board now. For large scale production of biomass, we require what is called a fermenter vessel or bioreactor. This is a simple diagram showing what we have, that is a fermenter vessel or bioreactor. See here, just like I mentioned about a washing machine, it is rotating constantly. So this is the agitator. It is normally just agitating the materials inside. Now the mixed biomass with the steam is allowed to enter inside. Aeration is provided with the help of aerator that provides oxygen, because with the help of oxygen only we can get what is the biomass. Otherwise fermentation just results in the formation of some other materials. Now we have just an outlet for cooling water to go out and also the products. This is a vent, a drain or we have or just overflow vent. This is what is called actually drain or overflow vent. And inside which we have a temperature sensor and also pH sensor. So they are sensing the temperature. They need an optimum temperature and also the pH, normal pH. It should not exceed, it should not be reduced, like the temperature also. So when it is normally just rotating, this is what is called the agitator. The materials are mixed and are produced in large amounts. Then we have the streaming, etc. other products. But here we have to know the simple picture what I am showing, that one is nothing but the fermenter or bioreactor. I'll give you a better picture just in the video. You can see that one, all these things beautifully. Okay. Now that's about the fermenter. So a fermenter is nothing but actually, you see that one, a closed vessel with adequate arrangements for aeration, providing oxygen, agitation, just for mixing, as in the case of mixing, temperature, pH control, and also what we have and drain or overflow vent, that is outside opening for just opening out to remove waste biomass of what is called the cultured microorganisms. Okay, cultured microorganisms along with their products, the resultant products, what we need to get. So afterwards we have streaming and then other process also. That's not necessary here. We have to know only what is a bioreactor, a fermented vessel, what is its use. It is for the production of, that is, the biomass in large scale. Before starting the industrial products, we have to make the differences between these three, what we have the words, that is prebiotics, probiotics, antibiotics. Now what do you mean by prebiotics? These are compounds, particularly the cellulosic fiber, normally found in food. That's main function is to induce the growth or activity of beneficial microorganisms in the intestinal tract. So the intestinal flora, the gut flora is being stimulated to grow and maintain their growth. That is because of what we have the prebiotics. So these are compounds in food, non-living substance that induce the growth or activity of beneficial microorganisms. Now what do you mean by probiotics? We have seen already about lactic acid bacteria that is probiotics. These are living microorganisms, not actually the dead matter, living microorganisms which intend to provide health benefits when consumed. How? Generally by improving or restoring gut flora. The gut flora growth is maintained. This is because of these living microorganisms. Even E. coli is also useful to just maintain the gut flora. And here I mentioned lactic acid bacillus. Lactic acid bacteria. So their main function is to improve or just actually restore the growth of what is called actually gut flora. Now what are antibiotics? So normally we can say antibiotics means against life. Anti-bio, working against life. That's why the name is given antibiotics. Normally we can say setting one thief against another. See the meaning for the setting one thief against another. We are producing some materials produced by the microorganisms which are detrimental or harmful to other microorganisms. So with the help of one microorganism we are killing another microorganism. That's why I say setting a thief against another thief. That is the meaning for that one. So these are all chemical substances produced by some microorganisms which normally kill or inhibit or retard the growth of other disease-causing microorganisms. For example, we have streptomycin. It is an antibiotic which is used to kill the bacterium, the one which causes tuberculosis. Mycobacterium tuberculosis, that's the bacteria which causes tuberculosis, that can be killed by means of streptomycin. The one which is produced by Streptomycin graeseus. So antibiotics are substances produced by some microorganisms which are harmful to other microorganisms. They are either just actually killing the microorganisms or inhibiting or retarding their growth. These are the two ways they are doing that, either killing or just inhibit the growth, thereby preventing their growth inside the body and preventing their effect to produce the disease. That's about the discrimination between these three, prebiotics, probiotics and antibiotics. Now we have to go for the microbes in industrial products. The first one we have to discuss about antibiotics. It's the first product because of actually industrial means we are producing that is what's called antibiotics. So I'll take the first one under the heading microbes and industrial products, the antibiotics. Okay, let us start with antibiotics. Under the heading microbes and industrial products, the first product we are taking antibiotics. I already given the definition for antibiotics. So these are chemical substances produced by some microorganisms which are detrimental or harmful to other microorganisms, either by killing or retarding the growth of other disease-causing microorganisms, even in low concentration. So that's why the name is given antibiotics. So antibiotics means against life. As they are working against other microorganisms' life, the name is given antibiotics. Antibiosis, that is nothing but the activity of what is called antibiotics to kill the microorganism. That is another one, antibiosis, the process. The activity of microorganisms to kill, that is what we have the microorganisms. The activity of antibiotics to kill other microorganisms is called actually antibiosis, otherwise called amensalism. I will come back to this one later. Now, the antibiotics are used to treat various diseases. A number of diseases being treated. Here in the book given only a few. For example, diphtheria, corynebacterium, that is actually corynebacteria diphtheriae. That's the one which causes diphtheria in the case of children. Then tuberculosis is a very common disease both in children as well as in the case of adults. Then meningitis, the inflammation of the meninges resulting in brain fever. Then we have syphilis, a sexually transmitted disease. We have seen already the plague, Yersinia pestis, the one which causes plague disease. Black death disease, otherwise called leprosy, a social disease. So these are all being normally treated with the help of antibiotics. Some examples for them. Now it was Salman Waxman discovered streptomycin, isolated streptomycin from Streptomyces aureus. Streptomyces gracius, not aureus. Streptomyces gracius. So he isolated what we have streptomycin, and he was the first one to use the term antibiotic. And he was the first man to use the term antibiotics. Now let's go for just what we have the discovery of, that is Alexander Fleming, the main miracle drug, the queen of drugs, what we have the penicillin. Okay, now let's see now. While working on staphylococci, just actually Alexander Fleming, normally he just working on staphylococci to find out the causative agent for pneumonia, how to cure it. But luckily he got the appearance of certain structures, and he found later it was a chemical substance. Due to that only he had certain structures appeared in the cultured plate, and he called it as a penicillin. So penicillin, while working on staphylococci bacteria, pneumococcus bacteria, staphylococci bacteria, Alexander Fleming observed certain green mold. Penicillin is produced by green mold, a fungus, found in one of the unwashed cultured plates, surrounding which the staphylococci could not grow. The meaning for that one, that chemical just only killed the microorganism staphylococci. So he found that it was due to the chemical from the mold, the green mold. That chemical only didn't allow the staphylococci to grow and killed all the staphylococci. And he called that chemical as penicillin. So it was the first antibiotic discovered by Alexander Fleming in 1926 as per the book, but it's actually 1928. So anyway, it is given in the book as 1926. Now, what was the causative organism he found? Later he found that it was because of the fungi, genus, what we have Penicillium notatum and Penicillium chrysogenum. So the Penicillium, the genus, produces a chemical, hence he called that one as penicillin, the product of what is called Penicillium, the genus of fungus. Now let's see what are the different actually genera. responsible for the production of penicillin. And what about the effect of penicillin when it was normally known about the effect of penicillin? So Alexander Fleming discovered penicillin. He didn't know much about the effect of penicillin. The effect of penicillin as a potential antibiotic was known much later only. Okay, let's see now the persons involved and the organisms involved, the fungi in the production of penicillin. Let's go further about penicillin. Some more points. Now, what are the genera normally produce penicillin? It is produced mainly by the fungi, two genera, one genus with the two species, Penicillium notatum and Penicillium chrysogenum. These two are green fungi or green mold. What is the effect of penicillin? It is a broad spectrum bactericidal in action. A broad spectrum bactericidal in action. So we can classify normally the effect of antibiotics into two groups based on the action. One is called bactericidal activity, another one bacteriostatic activity. In bactericidal activity, the antibiotic kills the microorganism. In a bacteriostatic activity, the antibiotics normally inhibit the growth of microorganisms by inhibiting one of the activities. Say an example, protein synthesis, etc. So bactericidal, killing the bacteria, bacteriostatic, not killing the bacteria but preventing or inhibiting the growth of the bacteria by blocking or inhibiting one of the events happening, that is metabolism, protein synthesis. Thereby the bacteria could be killed. Now it is a broad spectrum bactericidal in action which prevents the bacterial cell wall synthesis. Bacterial cell wall synthesis. So cell wall is the armor for actually the bacterium. When the cell wall being destroyed, automatically the bacteria will die. So the main function of this one is bactericidal, killing the bacteria. How? The mechanism, that is by preventing or inhibiting the cell wall synthesis, the outer covering of the bacterium, a protective cover. Once a cell wall has been removed, the bacteria are unable to grow further, unable to undergo cell division. So normally, this one, penicillin, is called queen of drugs. A wonder drug also we can say. Queen of drugs or wonder drugs. So its full potential is effective antibiotic. So normally just we see that one Alexander Fleming discovered that antibiotic. But the efficiency of that antibiotic, the efficiency, the potentiality of that antibiotic was established by two different people. One Howard Florey, another one Ernst Chain. These were the two people who established the effective activity, the potential activity of antibiotic. While they worked with the soldiers, while they treated the soldiers during the Second World War who were wounded, with the penicillin they treated those patients, and that's why the working ability, the efficiency of penicillin was actually opened out or revealed out. And for that, the three people, Fleming, Florey and Chain, they were awarded Nobel Prize in the year 1945 for their discovery of penicillin. So these were the three people who were awarded Nobel Prize in 1945 for their discovery of penicillin. That's about penicillin. Now we have to go for just actually some other antibiotics. And what is antibiosis? What are the types of antibiotics? Okay, let's see now. What is antibiosis? It's a process. It's an activity. The activity of antibiotics to kill microorganisms is called antibiosis. It's also called amensalism, a kind of animal association or simply association of organisms. Animal association used in medicine or association used in medicine. So we have different types of association just like mutualism, commensalism, etc. Antibiosis is otherwise called amensalism, a kind of association used in medicine. That was the neat question. Now what are the types of antibiotics based on their activity? That is a classification of antibiotics. One, broad spectrum antibiotics, another one that is narrow spectrum antibiotics. So what are broad spectrum antibiotics? So these antibiotics are active against a wide range of disease causing pathogenic bacteria. Disease causing bacteria or pathogenic bacteria. They can have the ability to attack major types of what we have disease causing bacteria. The narrow spectrum normally they are active against a selected group of bacterial types, not against many bacteria, only selected group of what is called bacterial types. This is about what we have the classification. Now we have to go for a few antibiotics examples. What is the role, effect, etc. I want to give only a few things only as per the book. A single paragraph is given in the book. After that we have to complete antibiotic resistance under the paragraph. Okay. Now let us discuss the activity of certain antibiotics. Number one, tetracycline. It is a broad spectrum antibiotic. It is bacteriostatic in nature or in action. What do you mean by bacteriostatic? Bacteria that limit the growth of bacteria, not killing, that limit the growth. Now one such example under the tetracycline, chlorotetracycline, otherwise called aureomycin. It is not in the book, otherwise called aureomycin. It was the first antibiotic of this group obtained from a genus Streptomycin. The species Aureophaciens, that's why it's called aureomycin. So chlorotetracycline, the new name for that one, the old name for that one, aureomycin. Now the next one, streptomycin. You know it was known by Selman Waxman. It is a broad spectrum antibiotic. It is a bactericidal antibiotic. What's the meaning? Antibiotics that kill bacteria. And that one just limit the growth of bacteria. This one kill bacteria. It is obtained from Streptomyces graeseus, working against both the gram-positive and negative bacteria, especially the tuberculosis bacterium, Mycobacterium tuberculosis. That's about streptomycin. And some other antibiotics isolated are erythromycin, chloromycetin, from again a species Streptomyces venezuelae, the neomycin, kanamycin, griseofulvin, bacitracin. These are some other antibiotics obtained from the microorganisms. Okay. Now the last one in the antibiotic, we have to discuss what is antibiotic resistance. A paragraph. Antibiotics should be used only when prescribed by certified health professional, otherwise we cannot take it. Now normally there is a question. Narrow spectrum antibiotics are preferred over broad spectrum antibiotics. Why? What's the reason? Because they are effective. The narrow spectrum antibiotics are effective and accurately target specific pathogenic organisms. They are targeting the specific pathogenic organisms when compared to broad spectrum. They are quick enough, but actually they are not specific. The second one, they are less likely to cause resistance. They are less likely to cause resistance. So they are just increasing what is the resistance power of the organism, that is the humans, but reducing the resistance of what is called the bacteria against antibiotics. Now, what is a superbug? So it is a term used to a group of strains of bacteria that are resistant to most commonly used antibiotics today. So many bacteria are resistant to antibiotics what we are using today. Such bacterial strains are collectively called superbug. It is a term used to describe the strains of bacteria that are resistant to most commonly used antibiotics today. So many resistant varieties are developed because of the human activities, or I can say anthropogenic evolution. Okay, so one more question I want to just complete. Now we are using penicillin. What is the problem associated with the use of penicillin? Just a few lines about this one. You see that one, the use of penicillin causes much problem. What problem? Hypersensitivity reaction is a major problem. Some people are hypersensitive to the penicillin injection. So hypersensitivity reaction is a major problem in the use of penicillin because it results in nausea, vomiting, wheezing, even cardiovascular collapse. That's why it's always better to have a sensitivity test before the administration of penicillin. So furthermore, doctors using a needle to prick a place in the forearm and injecting just that is a low dose, a big dose of penicillin. If any red itchy region appears, then we can indicate or we can conclude that that patient has allergic to penicillin. Okay, I write down on the board what is the problem associated with the use of penicillin, only 2 or 3 lines. As I discussed earlier, what is the problem associated with the use of penicillin? I mentioned hypersensitivity reaction is a major problem associated with the use of penicillin, resulting in some symptoms: nausea, vomiting, wheezing, and even cardiovascular collapse leading to death. So, sensitivity test for penicillin should be taken before the injection of penicillin for the patient. This is the one what I described. Now I just wrote on the board, so I just go through that one. So this is about antibiotics, one of the what is called the one of the what is called industrial products. We have to go for the next one, the beverages. What are the different types of beverages prepared? Mainly what we call the alcoholic beverages. The next class. Thank you.